Hey guys, let's talk about the adapted or acquired immune response to things. Remember, your immune system has two different types of responses. You have the innate or the non-specific immunity or immune response, sometimes called the acute inflammatory response. Think of like getting a cut or like a paper cut or a splinter or sprain an ankle, whatever. You got this non-specific response to things, but what we're going to take a look at is what's called the acquired or the adaptive response. And as we go through this, you'll see why it's called an acquired or an adaptive response. And really, it's made up of two different pathways for immunity. There's the cell-mediated or cell-to-cell immunity, and then there's the antibody mediated, or sometimes called the humoral immunity. Because humoral just means it's found in the humor or the watery environments within your body, like fluids and things like that. So we're going to take a look at these two pathways, the cell-mediated and the antibody-mediated, which both fall under the heading of this acquired or adaptive immunity.
Activation of the immune response typically begins when a pathogen enters the body.
Okay, so this pathogen they show here is just this little, red candy looking thing. But understand a pathogen could be a virus, could be a bacteria, could be a fungal or a protist. Anything, a pathogen is just a general term meaning something that can make you sick or cause an immune response.
Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces.
Okay, so the first player involved in this response, a lot of times are called macrophages. Macros means big, phage means to eat, or phagocytize, engulf, as you saw in the animation here. If you watch this, he just engulfs it, the membrane extends around it and brings it in like a vesicle. It doesn't show, but there are lysosomes that become involved, and they dump their digestive enzymes and breaks this pathogen down and a little bit of fragments, protein fragments. And what this thing does, it just sticks it out on the surface, says, hey, yo, this is the bad guy that's in here. This is what it looks like. And they call this the antigen presenting cell or the antigen presenting macrophage. So that's the first role player in this whole idea. And a lot of times from this point on, it sets off a cascade or chain of events that could go one of two ways.
Fragments on their cell surfaces. Macrophages.
Okay, now time out. It pointed at the antigen here. That's what these little red things are. So these are little protein fragments, little tags. Think of kind of like a wanted ad saying, hey, this is what the bad guy looks like. Be looking for him. If you see a cell or any with these on the surface or anything floating around that has this same look to it molecularly, because we're talking about molecular shape here and recognition identification, then you need to attack it, all right, and get rid of it.
Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells.
There you go.
An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen.
Okay, if you heard that verb there, it recognizes that antigen. We have little cells called T helper cells. T stands for thymus, because that's where they're created, in the thymus gland located on top of the heart. So you have these T cells, actually they're called T lymphocytes, and they have certain, receptors on the surface of the membrane that will actually recognize and dock in with this specific antigens being exposed by this presenting cell, this macrophage that initially digested the pathogen, broke it up and said, all right, fellas, this is who we're looking for. All right. So the T helper cell comes along first, checks it out, recognizes it, and you're going to see some stuff going on here with some chemicals. So pay attention to this.
During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2.
Okay, time out. We just witnessed a cascade. Similar to hormone functioning, when this thing connected, it released interleukin-1, which is just a type of protein that is going to be, or it might even be some are prostaglandins, I believe, and they just communicate back and forth and say, hey, this isn't supposed to be here. I mean, I'm personifying this thing, but this isn't supposed to be here, and then that stimulates the production of interleukin-2, another chemical molecule that's going to be released out by the T helper cells, and you'll see what happens next.
Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells.
Proliferation, as you're going to see in the animation here, just means it starts reproducing and making more of themselves, start cloning themselves. So these cytotoxic T cells and then these B cells, and B cells are created in the bone marrow. Right? They're a type of lymphocyte created in the bone marrow. So again, they're going, and that's where the B comes from. They're going to start proliferating, new term, just means dividing and cloning themselves.
The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells.
So this is where I'm talking about. We have cell-mediated immunity and we have antibody or humoral mediated immunity. Again, the cell-mediated immunity, as you're going to see here, is how we deal with cells that become infected. If a cell has been infected by a bacteria or a virus or a protist or a fungus or whatever, they're fungi, then we're going to use the cytotoxic T cells and what you're getting ready to see to deal with those. If we've got infected tissues and cells, we're going to use cells to destroy them. So we get cell-to-cell immunity. If they're just free, if there are pathogens floating around that maybe haven't made it into a cell to infect it yet, that haven't caused any damage, they're just floating around maybe in the blood or in the interstitial fluids between cells and tissues, then we're going to use what's called the antibody mediated or the humoral response to identify, tag, and remove those things that before they can infect another cell.
Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces.
Okay, so they're infected, they're sick, they're not feeling good, but in that process, they do have lysosomes as well that can also digest them, and they also have the ability to present those antigens. So basically, it's like a mercy flag saying, "Hey guys, I need help." You know, there's some bad guys in here. It's kind of like on our doors, we have these red and green cards. If there's an intruder in our room, we're supposed to put the red card up there. And if everything's good, we put the green card. So you imagine they're putting red cards up saying, "Hey, there's trouble inside of me. I need help."
The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell.
Okay, like I said, we have cytotoxic T cells that stay in circulation for a long time that are specific because you can see on the animation they have to have a specific shape to bind and recognize a specific antigen from a foreign pathogen. So if you get the current flu virus, you're going to have some cells, and some of your cells get infected with it. You're going to have some cells, cytotoxic killers, they're going to be floating around with a specific receptor that binds to that flu strain, that flu antigen. And should you get contact or get that infection again, then those cells will be ready and waiting, and it'll be a faster immune response. This is why it's called an adapted response or an acquired, because you don't acquire it until you become infected by it.
Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to
Okay, they just now jumped from cell-mediated into the antibody or humoral-mediated response. So remember, cell-mediated, if there's an infected cell that needs to be destroyed, we'll use cytotoxic T cells to come along. So we'll use a cell to destroy a cell. All right. This is a situation where there are pathogens floating around that haven't made it into a cell yet to do any damage or any infecting, but we still got to get rid of them because if we don't, then they will make it into our cells, and then we'll have to do cause more cells to die using cell-mediated, and if it gets out of hand, then we're in trouble.
Recognize a particular antigen. When B cells become activated by T helper cells.
So again, T helper cells, not only do they activate the cytotoxic killers, they also activate the B cells, and you'll see what happens next.
They differentiate into plasma cells.
So differentiate, they undergo some changes, and they're no longer called B cells, they're called plasma cells. And watch what happens next. And also they proliferate, they reproduce and clone themselves.
These plasma cells become antibody-producing factories.
And remember, antibodies are these Y-shaped proteins that have a specific receptor on them that will bind to antigens.
Flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens.
Okay, so if this was a bacteria or a virus or a fungus or a protist, whatever, all organisms have molecular tags on the surface of their cell membranes. And those tags will hopefully be recognized as being foreign and therefore elicit an immune response if they're dangerous to us. Right now, we have domestic bacteria in our gut flora and things like that are supposed to be there, but we're talking about things that aren't supposed to be there. So this little red lifesaver looking thing represents the pathogen and the antigen. Sometimes antigen and pathogen are used interchangeably. Antigen, it refers to the protein marker, the identifying tag that helps our body identify as it either belonging or not belonging. In this case, it doesn't belong. So these antibodies have the ability to attach to these antigens, and basically it marks them and says, hey, you don't belong, you don't belong, and that's going to help these macrophages are going to come along here to see them, because without the antibodies, these guys are invisible to our macrophages, right, and our other neutrophils and things like that that help digest and destroy things. So again, those antibodies are important because if our macrophages can't see or recognize that they don't belong, they won't engulf them, they won't destroy them. So again, antibodies are necessary because they mark the pathogens that are floating around. They haven't made it into a cell yet, but they mark the pathogens that are floating around, those foreign antigens, for destruction. They mark them so that we know to destroy them, and then along comes your big macrophages and other neutrophils that engulf it and destroy it.
Marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades.
Okay, so just waiting in the wings in the shadows, you have these other B cells that they don't turn into plasma cells to make antibodies. Instead, they turn into memory B cells. They have these receptors that will recognize that same antigen should it ever come back into your body again. And the benefit of this is that it helps create a faster, speedier, stronger immune response. So therefore, what may have knocked you out and made you sick and miss school or whatever for 2 days last time, if you contract the same thing, it may only manifest as a sniffle before your body has time to destroy and get rid of it, and you may not even realize you were that sick or that you had the same infection or the same pathogen floating around. And it's all because these memory cells are floating around and they're just ready. They're just waiting to turn into plasma cells and start cranking out antibodies so we can start marking those things before they get out and about and start infecting our cells and tissues.
Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
All right, guys, I hope that was helpful. I hope it clarified some things, helped you understand the two types of acquired or adaptive immunity. Like I said, cell-mediated, where if you have an infected cell, it's going to the helper T cells activate the cytotoxic killers, and they go around looking for the exposed antigen, the signs on the surface of the sick cells saying, hey, we need help, and they destroy that. Or we had the antibody mediator, or sometimes called humoral response, when you have these pathogens that are floating around and they haven't quite made it into a cell yet, but they have the potential to infect, and we got to get rid of those too. So we start producing antibodies that molecularly attach and match up with the antigens, those foreign pathogens that are floating around. And now all of a sudden they become visible to our macrophages and other neutrophils and other big eating cells that come around, and they just start chomping and nomming on all the little pathogens that shouldn't be there. I hope this was helpful. If you have any questions, you know where to find me.